Okay, I think we are ready to go. So we got about halfway, pretty close, through chapter 8. So we're gonna wrap it up today. Uh, I left off almost getting through aerobic respiration. We left off at the electron transport chain where we see the bulk payoff. So we talked hang on. We talked about glycolysis. We talked about citric acid or Krebs cycle. And here we are. So as we get to the point of the electron transport chain, when we think about this process, we have already dealt with that glucose molecule. We've blown it apart. We've liberated all of the carbon dioxides, and we've trapped those hydrogens on the electron carriers. And now it's the job of those electron carriers to dump their cargo off. That's the idea. So what we're looking at here is bacteria, but we can also think of this as something happening in our cells within the mitochondria. Same sort of an idea. And as we've described with the endosymbiosis, some of these little ancestral aerobic microbes were picked up by some other microbes and ultimately kept around. So we'll think more about that a little later. So what we're looking at is just a little fragment of a bacterium, and we're looking at the plasma membrane. All right. So up above it says outside of the cytoplasmic membrane. What's outside of the cytoplasmic membrane? I heard one little whisper. What do you think's outside of this with our bacteria? So silent. It's a cell wall, right? Are you shaking your head? Are you like, that's what I said? Okay. So the peptidoglycan, right? And it depends on the type of bacteria. If we're talking gram negative or gram positive for what we truly are seeing outside of that. But the cell wall is truly the first thing that is out of that. And then if we're talking a gram negative, after the cell wall, the peptidoglycan layer, there's an outer membrane, all right? But it's the space in between the cytoplasmic membrane and the cell wall that becomes important here, all right? When we think mitochondria, it's the outer membrane of the mitochondria, and the inner membrane, we call that space the inner membrane space, all right? This, as we get into this area, we just say extracellular space. Same idea though, all right? So all of the process of breaking down those sugars, going through citric acid, all of this stuff is happening in the cytoplasm of our bacteria. It's happening in the matrix of a mitochondria, the inside, all right? And once we get all of those hydrogens loaded up on the electron carriers, now they're going to start releasing those hydrogens. So we see a handful of purple channels within this membrane. These are some transport channels that are going to allow the hydrogens to move through the membrane, active transport like, as we are getting pushed into this extracellular space. We're getting pushed against a concentration gradient. We're accumulating boatloads of hydrogens within this space, like putting a bunch of water behind a dam, right? Now you've got the force behind this, a huge amount of potential energy that can trickle through ATP synthase, our pear-shaped protein enzyme in the membrane, and they move by diffusion from high to low concentration. And the energy from these hydrogen ions, as they're percolating through there, is enough to recharge the ADP molecules, putting a phosphate on there, recharging them. Yes?
So does the hydrogen get recycled? It's like being pushed out and then being absorbed back.
That's a super good question. What do we do with the hydrogens? What happens to all of that hydrogen, right? So as we are percolating through, we're dumping off with our carriers hydrogens and shoving them in here, all right? Through all of these carriers, the red stuff is electrons. So it's the energy that's moving through there, and we're like active transport. It needs some energy to do this. Hydrogens that percolate back through there, we're not this red arrow, there should be a little H down here, H plus, it's coming in here, right? So the hydrogens move in, they ultimately participate over here, all right? So recycling, yeah, it's like recycling. These hydrogen ions are final, when we're talking aerobic respiration, our final protein over here is cytochrome oxidase, cytochrome C oxidase. It's our last bit, and the energy from that electron, similar, not the same, but similar idea when we think about photosynthesis and the photons bombarding down onto a plant or onto a photosynthetic bacterium. We start exciting stuff, right? And that energy can be utilized to do something, all right? So this energy, as it's moving through, we've shoved hydrogens, but this last little bit, it's got to do something. And when I think about energy, whether it's through photosynthesis or those electrons that are moving around, it reminds me of being little and dragging my stocking feet across the rug and getting a boatload of static electricity. You could do the same with a balloon and rub it on your hair and stick it to something, right? But you could charge yourself up and then shock somebody. That was always fun, right? So that shock, that charged up energy, that's this red arrow as it's accumulating within you. Now you've got this energy. What are you going to do with it, right? That energy shocks something. The something it's shocking here isn't you, it's shocking oxygen, all right? O2 molecules, aerobic, we want oxygen. As we are pulling oxygen into our cells, we break them apart. O2, atmospheric oxygen, gets blown apart, and oxygens would like to participate in some bonding. They'd like to, if they could, or oxygen gas double bonded with themselves. So two hydrogens link up, and we show 1/2 O2 to signify we're talking atmospheric oxygen. That's what we're talking about, all right? So two of these come into the mix. Half of that oxygen gets linked up with this. Technically, we should show both of it, 4 H pluses and 2 waters from a single O2 molecule that gets blown apart. We're only showing you half of it, all right? So they get recycled, the hydrogens, onto oxygens. And those waters now, they could participate in other things, right? In cellular respiration, water is a byproduct. And in photosynthesis, water is an input, right? So we're generating this, and photosynthesizers pulling it in, not directly from us, but they're pulling in water molecules and they're splitting them. releasing out oxygens that we pull in. It's a cycle, right? We need both, right? So as this is happening, you can see all of these carriers, they have, at this point, with their hydrogens on them, they are in the reduced form. They're loaded up with their cargo, and they get oxidized. They dump it off and go back to the oxidized state and are ready to get more hydrogen slapped onto them. They quickly have a turnover with this, picking up hydrogens and dumping them off, all right? And as we're doing this, constantly cycling through, pumping hydrogens, making waters, deep down, making energy. That's what it's all about, all right? So the bacteria, the aerobic ones are doing this when we're thinking about cellular respiration, aerobic, but there's other types of respiration, and truly the first types of cellular respiration weren't aerobic, they were anaerobic. And there were different, and there still are, organisms that utilize these mechanisms, but they don't tolerate oxygen. They use somebody else, hydrogens or sulfurs, and they split these molecules and start dumping hydrogens onto them. All right. That in turn, same idea for bulk payout of energy. It's just somebody that's the final acceptor that's different. All right. Not an oxygen, making it anaerobic. All right. But everything else, same sort of an idea. All right. There are others that can't go through all of these pathways. And then we start thinking about fermentation mechanisms to go through and generate minuscule amounts of energy because they lack all of this stuff. All right. They don't do it. And there are others that have it, but also can ferment if oxygen gets depleted. Our own muscle cells go through this process and generate lactic acid when they become starved of some oxygen. All right. So we see our bacterial cell with its cell wall and with that endosymbiosis theory, as these cells were engulfed by other cells, endocytized, they are pulled in, and now they are within a vesicle that's part of that host cell. Ultimately, the cell wall material goes away, and we are left with plasma membrane from that bacterium and the vesicle that got brought in, a double membrane bound organelle, functional mitochondria, right? Which, when I think of all of this and the microbes doing all of these reactions, and then I think about the mitochondria, it makes me kind of stop and wonder, are we just some vessel for these mitochondria to ensure that they've got an ample food source? Are we just shoveling it in because that's what the mitochondria truly want? Are they the ones that are driving us, right? Hard to say. Kind of makes you stop and think just the littlest bit, right? Who really is in control here, right? What forced me to have that donut for breakfast, right? Was it some of these microbes? Was it my mitochondria? Was it my microbiota that lives in my gut? What the hell's going on here, right? All right. I like that, all right? I feel like I've got some sort of control, right? But then I wonder deep down, do I? Right? What's happening here? All right. So this recaps what we've discussed so far as we move through here, starting off with glycolysis or the Embden-Meyerhoff-Pranis pathway, pulling in glucoses, generating pyruvates, and showing what do we get as we do this stuff. So energy is really what we're thinking about But the electron carriers over here are really the source of generating this stuff. So we don't want to disregard these guys as not really being important when we're thinking about, oh, well, look how much energy we get. We need these to establish that concentration in the first place. So we get some hydrogens loaded up. We reduce some NAD up here. We make a little bit of energy. And like I said, with some of those that don't do any sort of respiration, this might be as far as you go. That's not much energy. All right. And when you think about that, bacteria that are solely fermenters and don't have any sort of respiratory pathways typically grow considerably slower than organisms that do have the potential for some sort of respiration. They get a lot more energy. It should make sense. If you're only making a minuscule amount of energy, you're not going to grow very rapidly. All right. And we'll talk similar stuff like that with some fermentation as well, because we see typically both aerobic and then anaerobic, and there are shifts in the growth of those cells, whatever they may be. All right. So the net and theoretical yields that we can see as we move through here, and recall as we think theoretical stuff, this is if we pulled in that molecule and completely blew it apart and used every single piece of that to drive the production of ATP. But we're building all sorts of stuff. So as we're shuttling through this, we're not just going through and completely catabolizing all of that. Some of it drives off into anabolic reactions, building stuff. So cells typically aren't really achieving the maximal yields because they're doing other things as well. All right. But they could, in the perfect situation, produce boatloads. All right. So the next bit from glycolysis is this transition reaction. This is what's preparatory to the citric acid cycle. We pop off one of those carbon dioxides, creating a 2-carbon acetyl group. That links up with the coenzyme A, forming acetyl-CoA, and that shuttles into the Krebs cycle. If we're thinking mitochondria, that's what helps to facilitate those acetyl groups to move from the cytoplasm into the mitochondria. All right. But in a bacterium, it just shuttles it over into the citric acid cycle. It's already in the cytoplasm. That's where everything's taken place, right? So then to Krebs, those acetyls get degraded, all right, the 2-carbon molecules. So you blow off the rest of the carbon dioxide, all right? As we left off last time, this was the end bit. We had no more carbons left from our glucose that we pulled in. And all we have done now is link up these electron carriers with their hydrogens to start the payoff. All right. That's the idea. And total amounts as we move through there. Those hydrogens, and this at the end is if, you know, we're going through that transport after all of this happens. So through phosphorylation, all right, oxidative phosphorylation, the electron transport chain. So for fermentations, it really depends on circumstances. There are definitely opportunities where organisms don't have potentials for respiration. So all you get is this. It's just fermentation, right? But typically, when we think about cells, like the production of ethanol makes me think about yeast cells, all right? Some yeasts super good at producing alcohols, all right? Bacteria, some of them also have potentials to do this. They can ferment and produce other things as well. All right. Depends on the circumstances. All right. And through a lot of different fermentation pathways, numerous things, we rely on microbes for their generation. All right. So if you eat some yogurt, maybe you had yogurt for breakfast instead of that donut, right? Yogurt is produced through the fermentation of lactose from some lactic acid bacteria. And truly, there are handfuls. If you look at yogurts, you will see all sorts of different microbes listed on the packaging, right? A lot of different things, all right? So we'll think a little bit about that with lactic acid being generated. But first, we want to think alcohol, all right? This is the littlest bit different in this process because the pyruvic acid, the 3-carbon molecule, gets processed the littlest bit. And when we think about lactic acid fermentation, we just manipulate it. We're not going to lose a carbon dioxide. All right. So lactic acid, still 3 carbons. All right. Ethanol, 2 carbons. All right. So the pyruvic acid, one CO2 comes off, and in the process of making alcoholic beverages, there's a lot of carbon dioxide that gets produced. So CO2 being released, the 2-carbon molecule that's the result of that is acetaldehyde. It serves as the final electron acceptor, and NADH dumps its cargo back onto this. All right. And as it donates that hydrogen to acetaldehyde, now slapping an extra hydrogen onto it, you've got an ethanol molecule. You just made alcohol. All right. So that's the idea with fermentation. And when it starts off thinking yeast cells and making some sort of alcoholic beverage, you start with something that is aerobic, and you would like to have ample amounts of oxygen so those yeast cells can start growing, feeding, replicating, so conditions are optimal. And they start growing very rapidly. And in the process of doing this, they're producing boatloads of CO2. They're fully respiring, all right? They're not making maybe a minuscule amount, but they're not really making any alcohol at this point. They are burning through that stuff. What would they be producing if they're not making alcohol? What's a byproduct? Cellular respiration. We just talked about it. So silent. Zoomers, I probably have you muted. Can't hear you. All right. Water, right? That's what they start making. Would you want to decide you're going to make yourself some sort of home brew? You're going to make some beer or some wine. Would you like to just leave that thing opened up and just let it rip through there and produce boatloads of CO2? What else are you producing? Boatloads of water. You're going to be left with something that's not so good, and definitely not something that's going to give you any sense of euphoria, right? Not going to work, right? So what do? We have to manipulate this stuff, right? We want to get these guys growing really good, and we put a cork on it, all right? Basically capping off the amount of oxygen that can get in there. There's still a little bit of residual oxygen in your vessel, all right? But you put an airlock on there. So you don't want to just cork it or you're going to have an explosion. You put an airlock. So something that allows gas to be released, but it can't come back in. So all of the CO2 now, still making CO2, all right? But it starts to accumulate up in that airspace, pushing out all of the oxygen, and what you're left with is anaerobic sort of environment. Boatload of CO2 because we just keep making it. Now we've gone anaerobic. And at this point, you can't go through the electron transport chain. There isn't any stinking oxygen left for this to happen. So we ferment. And then this vat of stuff that was bubbling super rapidly slows down. And you see little bubbles that are emerging out of there instead of like it's boiling stuff. All right. And it slows down because we're not generating as much energy. We're not growing as quick, and we're pumping out alcohols as a result of that. Right. That's the idea. All right. So you've got to create an anaerobic environment if you want this process to continue. All right. We can flip it around the other way and think about ourselves. And in the process of imbibing in some ethanol, as we start to break that down, the first thing that we start to produce is some acetaldehyde. This stuff isn't so good. And if you have enzymes that are faulty and you can't manipulate this acetaldehyde, imbibing in ethanol makes the acetaldehyde accumulate, and then you don't feel so good. All right. Typically, those people that have those faulty enzymes aren't imbibing in ethanol because they get super sick. All right. Not able to tolerate that stuff. And enzymes are truly what's the result of that. All right. So, and if you have functional enzymes, but you overload the system because you imbibed in way too much ethanol, all right, you're going to wake up the next day and probably not feel so good. Why? You got a lot of this stuff that's still needing to get manipulated, and it accumulates and you don't feel so good. All right. Some people would call that a hangover, right? Not so good, right? And that is truly what's happening inside of your body. Okay, I mentioned as we started this in the lab, you're doing a handful of different biochemical tests, all separate tubes, and you inoculate stuff to see what sort of things may they tolerate or may they use, potentially ferment and produce some acid product that you could detect. There's a lot of different things, right? And I mentioned that there are also some sort of rapid, you still need cultivation, but some rapid tests. So this is an API test, a rapid test. The idea with this, each of these little wells is basically like a separate test tube that you could be inoculating. There are some others, not APIs, Intero tubes. They look like it's a tube. They look like a little rainbow colored popsicle is sort of what I think. And as I'm looking at this, it's sort of rainbow colored as well. All right. The Intero tube has two caps on it. And one cap is a needle, like a loop, right? But it's just a little point on it. And the other end is the handle. So you unscrew the one with the needle, you touch a bacterial culture with that needle, you unscrew the other end to get the handle, and you start pulling it through the tube, twisting it, all right? And as you twist that rod up into, and it's, imagine that's what's happening here, all right? As it moves through here, you get to the very end and the tip of that needle is in the middle of your last well, and you shove that sucker back through there. All right. And as soon as the tip of it hits the first well, your handle's still going to be poking out, and it's got a notched little groove in it, and you snap it, cap them both up. And with the end that you snapped off, there are a few wells that you poke a few holes in because those are going to give you aerobic conditions. And the rest of your tube is sealed with some plastic, anaerobic stuff. Now we can test both simultaneously. All right. Same stuff happens with these API strips. It gets inoculated through the strip, and we don't see it, but a few of these will have some little vent holes that allow for gas exchange. Others not. All right. And I don't know all of these things on here. Half of them I can't even see. Uh, but different types of sugars. I see right here URE, the urease test. I think you are doing that right now, or you are next week. It's coming up. Urease test? Anybody? Yeah? On Thursday. All right. So this one, if this has been inoculated, I'd say it's a negative, right? If your urease test is positive, it looks kind of like this, super bright pink, all right? Kind of cool, all right? So regardless of the system, all right, we inoculate this stuff, we incubate it, and then we look for changes. And we've got a key that shows us what it looked like to begin with and what it would look like if it was a positive. And then you basically go through there and you code it out. So counting up the number of positives. And as you see here, I've got an arrow, an arrow, an arrow. So I group these first three. All right. And dependent upon the result with that, however many are positive or whatever, I get a specific number as I add them together. I move over into my next one and I count that up. And so when it's all said and done, I get a what, 1, 2, 3, 4, 5, 6, 7-digit code is what I get. All right. This code, this kit comes with a code manual, and I look up my number. And as I go through there, finding my number, right off beside it is a bacterial name. This thing matches this bacterium. Done deal. All right. That's pretty cool. All right. We used these things in the lab quite a few years ago. All right. Interrotubes and oxyferms were the ones we did, not the APIs. All right. The little popsicle looking guys. All right. We used these and then we stopped using them because they didn't work for us. And the reason for that is the bacteria that we play with. All right. If we were working in a clinical situation with real pathogenic microbes, my gut says this system would work much better. All right. Why it doesn't work so good for us is because most of the bacteria that we work with has been super attenuated. All right. When you attenuate a bacterium, you basically cultivate it repeatedly. All right. And as you keep this cultivation process happening, things happen to those microbes. They start losing some of their characteristics. And what we truly want them to lose are virulence factors, the potential to be more of a disease causing microbe. If you can throw that out the window, now you got somebody that's relatively safe to work with in a lab. That's what we would prefer, right? Rather than rolling in and being like, well, I just gave you all tuberculosis today. Sorry, right? You don't want that, right? You want to work with somebody that's not holding those potentials to maybe kill you, right? Not a good thing, right? So when we lose some of those factors, it ultimately impacts some of these biochemical tests and makes things potentially not function exactly like we want them to. So we'd grow up some E. coli and then run it through the test, and it would pop back as somebody completely different, right? But it looks like E. coli. Everything that you do, a few simple tests still point to E. coli, but you throw some of these extra ones in there and you get a variation, right? Where now this thing that should have been positive no longer is. Completely different code number, completely different species, but still E. coli, right? So we were using those things for unknown stuff, which makes it a big problem when you're dished out an unknown and they're like, tell us what this is, and you code it out and it's completely wrong. That's not so good, right? So we decided why, right? Why waste our time with that stuff? And they were also super expensive, right? So better off to do what we're doing right now. A few key biochemical tests. And for the most part, they do what we want them to do. All right. E. coli should ferment lactose. When you do those those phenol red broths with lactose in there, E. coli should make that turn yellow. And you also should see some gas bubbles in the Durham tube. All right. In those Entero tubes, there's a bit of wax over the media. And if it produces gas, it pushes that wax up so you can see gas was produced. All right. And it's a similar thing over here where you can see some bubbles. Maybe you can see some bubbles, but there's a couple of little bubbles in there. Gas production. All right. All right. Do I not have I don't have anything with our lactic acid fermentation. Now I swear I scrolled through this and I don't think I saw it before. Real quickly with this, when we think about bacteria like yogurt production, all right, this bit right here doesn't happen. All right. The pyruvic acid, as we go through glycolysis and make 2-3 carbon molecules of pyruvate or pyruvic acid, they are what serves as the final acceptor. And NADH bumps right up into the pyruvic acid and dumps their hydrogens off onto it. And now you've got this 3 carbon molecule with a hydrogen added to it. That is lactic acid. All right. That's what our bacteria are doing when they're making yogurts. All right. It's also what our muscle cells do as they are starved of oxygen. The mitochondria aren't able to aerobically respire as optimally as they should. And instead of doing all of that, the glucose gets split in half and still within the cytoplasm of your muscles, we dump hydrogens back onto those pyruvates and the lactic acid accumulates in your muscle cells. That's why you get sore. That's why you see a marathon runner making it almost to the end and they fall over. Their muscles freeze up. They try as you might. You can't take another step. All right. And deep down, it makes me stop and say, you shouldn't do that to yourself, right? Why run, right? Well, you could kick back on the couch with a frothy beverage and watch somebody else do it, right? Much better, right? Don't have too many though. You're going to feel it. All right. There you go with your yogurt. Probiotics, they're good for your gut, right? All right. I know there's some yogurt lovers out there. There's got to be. We want to think a bit about photosynthesis. And as I scrolled through this, my first thought was, why? We're sitting here in microbiology class. Should I be telling you about photosynthesis in the first place? Technically, we kind of need a little of that, right? We wouldn't be here without it. And in turn, microbes with some potentials, although this we're looking at a plant cell, but microbes with potentials to photosynthesize truly were the cells that started the whole process of the oxygen revolution and the reason why we're here in the first place, right? And I stopped and I'm like, it probably is worthy of discussing a little bit. All right. So these chloroplasts were once photosynthetic bacteria, just like the mitochondria. All right. Similar sort of an idea that got engulfed and kept around. Now you got a cell that pulled this thing in to eat it. And it's like, wait a minute, it's making food for me. Why would I destroy it when it's just producing foods for me? That's a beautiful thing, right? And I've already engulfed some mitochondria, so I can just shuttle those sugars right over to him, right? And there we go. Closed system. So as this happens, the energy from the sun, these photons are bombarding chlorophyll pigments that we see on these thioploid membranes that are stacked up together to make a bunch of discs. We call that granum. And they function together to harness as much energy as they possibly can. They technically have some other players involved. There's accessory pigments, carotenoids, that help to snag some extra energy that some of the chlorophyll pigments aren't quite able to grasp. All right. So in the process of doing this, we've split this in half. We've got light dependent and technically independent reactions over here, but we are relying on the fact that this happened in the first place. All right. So the photo portion, the dependent reactions happens on these membranes, exciting chlorophyll pigments, exciting electrons. All right. And that excitement, like the stocking feet, that energy gets harnessed, and we turn that energy to a water molecule, shocking the water, blowing the hydrogens off of it, and releasing oxygen gas. We snag those hydrogens on electron carriers, and we shuttle those guys next door. We shuttle them out into the stroma where these guys are floating. All right. So their carriers come out here and start interacting in the Calvin cycle, which is not the same as the citric acid cycle in reverse, but pretty much is. All right. It's like the opposite. All right. CO2s are being poured in to this cycle, and in the citric acid, we're shooting them out. Right. So the plant's pulling in CO2, it's using energy and those carriers. The hydrogens get dumped off, and we start manipulating them, building this molecule that will turn out to be glucose, all right, as we keep going. The organics coming off of here, glucoses, truly starting with simple fragments, right? Glyceraldehydes, things like that, all right? We start building these molecules, and just as we think about with respiration, with maximal yields, it's the same stuff happening over here. There are parts that get shot off for other pathways. It's not just flat out making sugar. All right. We gotta make the plant's making everything it needs, right? It's more than just sugars. All right. So the energy generated here only is driving this reaction. This energy isn't something that we think about that is driving other cellular work. That stuff comes from the mitochondria. That stuff's coming from the metabolism of their products. All right. And the cycle keeps going. All right. So, and there's all sorts of variation. We're not going to, so I'm not standing here teaching you botany. All right. But there's definitely variation when it comes to plants and how they can utilize the CO2 either in the heat of the day or potentially like a cactus opening up their stomata in the middle of the night and pulling in CO2 to reduce water loss. All right. All sorts of different potentials. That's not for here. All right. So for eukaryotes happening in the chloroplasts, all right, but in prokaryotes, a bit different. All right. The membrane that we see here, the membrane of that bacteria, the cytoplasmic membrane or plasma membrane, increases, enfolds into the cell. And just like the proteins studded on the membrane for electron transport, similar proteins studded on this membrane to start shuttling those hydrogens, right? We want to push them against a concentration gradient. And in these photosynthetic bacteria, it's the same sort of an idea. From the membrane in between space, and the cell wall. We want to shove all these hydrogens into this space, right? And then percolate them through and start ultimately making sugars, right? Is the idea. So with this, we've got all sorts of pigments. This light harvesting complex, when we think about chlorophyll pigments, there's two different chlorophyll pigments picking up subtle differences in the wavelengths of energy, and they function together, all right? So we start snagging some of it, and this they say is like an antenna system amplifying, all right, the energy as one picks it up to the next and so on. We start shuttling that. We get excited in this one, shuttle that energy to the other. It's already getting a little excited. Now it's got more energy and it's even more excited, and it keeps shuttling it. Finally, to our last one, the reaction center, it's going to take all of that energy that everybody else has accumulated, and it shoves it up into that reaction center. And this, it's not the same, but when I look at this, this makes me think of electrons orbiting around the nucleus of an atom. And when we think about atoms, their valence shell of electrons is what dictates if they participate in any bonding or not, right? And if you have an electron out there in this shell, you may want to do something, all right? The reaction center is basically like that, where it gets supercharged and that electron, that energy gets jolted up. They say a quantum level. It goes to an orbital that sucker doesn't want to be at, right? Now you're super reactive, and that energy is going to do something, all right? So it's going to split water apart is what it's going to do, all right? So we can see some of that energy, our electron in red, moving into electron transport chain, but down below, as we see this is where we start splitting, all right? And it depends, all right? If we're thinking photosynthesis, it's the same idea as I described with respiration. There's aerobic and there's anaerobic, all right? And for photosynthesis, same thing. There's oxygenic, aerobic, if you will. It's generating oxygen, and anoxic, no oxygen involved in this, all right? And the first photosynthesizers were anoxic. They were using other molecules than waters, all right? And they were generating different things. And then we transition to the potential for cells to utilize water. And that's the start of the oxygen revolution. But it starts off with photosynthesizers not using waters, not making oxygen. They're making something else. All right. Just depends. And that's why our A over here, because it doesn't have to be an oxygen. All right. It could be somebody else. Just depends on who we're talking about. All right. When we think about plants, it's an oxygen, right? And lots of photosynthetic bacteria, same thing, but a few of them, it's different, all right? So that's this stuff down below. The oxygenic, everything we just described, carbon dioxides and water coming in. We use the energy from the sun to blow the waters apart, and then we start putting hydrogens onto these CO2s. Calvin cycle grabs them and moves them around, all right? And the end result is a sugar, some oxygens, and some water. That's what you get through oxygenic photosynthesis. Anoxygenic, we've got somebody else besides water, hydrogen sulfide or hydrogen gas or other stuff, all right, whatever it might be, all right? And we go through this, building some sugars, right? Something else, not oxygen, right? And we also do generate some water, but we're not blowing water apart, all right? So water is still a byproduct, all right, but not necessarily an input, all right? Truly, depends on those circumstances. So we're just about out of time, but also just about done. So in plants, we've got two different photosynthesis systems. And this always is a bit frustrating to me. I teach non-majors biology class, and I like to say it's stuff like this that makes people hate biology, right? Photosystem 1 was discovered first. That's why it's called photosystem 1, but it happens second. And I'm like, that's just stupid, right? Why don't we call this 1 because it's first? And you can have a little footnote and say, it's the first thing, but we discovered it second. Deep down, I wonder, who really gives a crap when it was discovered, right? Somebody does, right? But that's the deal. It happens first, and you see different wavelengths with these chlorophyll pigments, 680 versus 700. Done deal. Shuttling energy, making ATPs, and producing sugar. All right, I will quit with that. We will talk about some nutrient cycling. I thought I was about done until I saw this, and I'm like, wait a minute, I've probably got 9 slides left. All right, farewell.